All right. Hello, everyone. Happy Monday. Thank you for being back. Thank you for your patience on Friday. Sorry, I haven't been feeling too great, but we're back. We're going to make our way through the GI system. So just wanted to show you some of your exam stats from last week. Looked great. Median was an 85. About 29% of you were in the A minus range. But a reminder for those of you who weren't so happy with your exam scores, we are in this class allowing one retake at the end of the semester during our scheduled final exam block, which will just follow the block that's assigned to us on the registrar website. I've got an email saying that we've got some different conflicting dates and some different documents. I'll check that those are all aligned, but we're just following what it says on the registrar's page. It's the sum total of that. Okay, so hopefully y'all took a look at the beginning of our functional anatomy of the GI system. So I got a couple questions for us so we can review that and touch base a little before we move into our new content. Okay, so one of the things that we were talking about were our layers of the walls of the GI tract. So this is going to be our sort of basic format for kind of any piece of our intestinal lining, but we'll be talking about specifics as we go through specific regions. Now we're going to start with what part of the GI tract is actually touching your food. Okay, great. Yeah. Okay, so the innermost layer coming into contact with anything you're eating or drinking is your mucosa, your mucosal layer. So that's touching what we refer to as the lumen of our GI tract. Perfect. All right. We might also have some absorptive cells we'll be interested in, especially as we get into our next packet, absorption. So let's think about where those would be or what those are. Let's take a look. Okay, cool. So enterocytes, we have a couple of classes of enterocytes. We're going to have our endocrine, our exocrine, and our absorptive cells in this category. They're part of our epithelial lining. So again, absorptive cells, we'll learn a lot about how we're absorbing nutrients using those. Okay. And one more before we move forward. Okay. So another part we had in here is you actually have sort of almost a branch of the nervous system embedded in your GI tract. And it's got kind of two different parts. So I want to key in on, did we notice where those two layers were, where we had nerves specific to the GI tract? Okay, great. Yeah, so this is going to be in our submucosa and our muscularis externa. Perfect. For those of you who didn't answer or a little confused, remember to go through your slides and your book where you see these images going through the layers of the wall of the GI tract and then breaking down specifically what's in each one of them. But here where we can sort of see these yellow fibers, those are the two parts of the enteric nervous system that we have here. So you can see them in that submucosal layer and this muscularis externa layer. Okay. So after that, we began walking through the path that we'd follow if we were a piece of food, right? So we started with the mouth, you're chewing. This is the beginning of our digestion process. Digestion, just breaking down what you eat. And we'll walk through the specific enzymes related to that again as we go through how you digest and absorb any given macromolecule. We're passing from our mouth into our pharynx, and then through our esophagus, right? And we're paying attention to our esophageal sphincters because some of them are under your control and some of them are not, as we have a part made of skeletal muscle, voluntary, and smooth muscle, which is involuntary. Comes back to, if we remember from A&P 1, how we have our somatic portion of the nervous system doing our skeletal muscle versus our smooth muscle being the responsibility of our autonomic nervous system. Okay, so we left off with the stomach. So after we go through the esophagus and we go through that lower esophageal sphincter, we find ourselves here. So we got this sort of J-shaped hollow cavity. That's our stomach. Got different parts to it. So up top, we see a fundus, then we see a body, an antrum, pylorus, and a sphincter here. You may have looked at these in lab. So this is where we're storing our food after it's swallowed. We got relatively small stomach if it's empty, can get real big after you eat some food, drink some water, because we've got all these folds or rugae that allow it to expand. Okay. So from a physiology perspective, one of the things that we're going to be interested in is how the stomach is actually helping us to break down food, and also a little bit of how we fight pathogens. So our gastric juices and our gastric pits become important here. So your stomach's making gastric juice. We'll go into the components in a second. But we're going to be mixing this gastric juice with the food that you've chewed, swallowed, okay? And we're going to use a new word for this new mixture that we've made. So this is your chyme, that chewed up kind of food and gastric juice smoothie, right? And it's then able to pass through our pylorus and our pyloric sphincter in little bits. We want to make sure that we've got not a huge ball of food all going into your small intestine at once, basically. So this is a control of our gastric emptying, basically. Okay. So let's take a peek at those gastric pits. Okay, so what we're looking at here in the gastric pits, right, we're zooming in on the tissue of the wall of the stomach here, this lining of the stomach, okay? And we've got these kind of holes going deeper into the tissue. And as we go down into a pit, you're going to go through a neck where we're going to have a bunch of mucus. And you're going to go into the deeper part of the pit here where we have our gastric glands. Okay, so these gastric glands, or these deep parts of the pit, are making stuff that we need for the stomach to function. Okay. So we're going to be paying attention to where these products come from. So I'm going to point out that we have these parietal cells in purple, and we have these chief cells in this kind of beigeish color as we're down in these deepest parts of our gastric pit, which we're calling gastric glands. Okay, so the different cells are going to make different things. What the chief cells are making, so these beige ones, right? Those chief cells are making something called pepsinogen. Okay. So pepsinogen, are going to notice some parts of this word. So this inogen ending we've seen before on some other molecules usually means we've got something that's going to need to be activated to function as an enzyme, which is the case for pepsinogen, just like it was when we were talking about our different clotting factors, for example. So pepsinogen going to be this precursor or inactive form of an enzyme called pepsin. And we can also notice that pepsin, or this pep beginning, looks a lot like peptide, right? Or polypeptides were those long chains of amino acids that fold into proteins. So pepsinogen is this inactive enzyme that's ultimately going to help us break down proteins. That's how it gets its name. Okay. So that's coming from our chief cells. So the chief is in charge of breaking down proteins. Okay. So our parietal cells are still important. We see fewer of them here, these purple ones, but they're going to be secreting two other important things for stomach function, two important products that we see in our gastric juices. So one of those is our hydrogen ions. So remember, hydrogen ions, H plus, right? Basically just a free proton. So it makes things acidic, right? When we have a pH, right, we're measuring how many free hydrogen ions do we have? How much acidity is there? So when we say stomach acid, right, we're talking about these hydrogen ions that are being produced by our parietal cells. So we're going to find that the stomach is super acidic. If you've had a stomach bug ever or recently, you may have noticed that acidity of the stomach contents, right? So both our gastric juices and the stomach acid, as well as then the chyme, when we mix together our gastric juices with some food, are going to be pretty acidic. And that's important both for the breaking down of proteins, as well as for hopefully killing off maybe some of the bacteria and stuff that might be on your food. So we're maintaining an acidic environment with these hydrogen ions coming from our parietal cells. The other thing that those parietal cells are secreting is something called intrinsic factor. So intrinsic factor, something that helps us absorb vitamins, specifically vitamin B12. Vitamin B12 we need for things like making new cells. So we need this intrinsic factor to make sure we have the components we need to keep our body working and healthy. Okay. So those are our chief cells and our parietal cells. We don't have our G cells pictured on this image from our textbook, but they are also important. They secrete something called gastrin. So gastrin is a hormone. So gastrin is going to get into the bloodstream, and this is going to be sending signals up to your brain and kind of throughout your body, telling you about the function of the stomach and what it's doing. Now that's happening down in the base of the gastric pit, all of these functions. But if we head back up to the neck, right, imagine we've got our stomach acid and the gastric juices containing all these other products coming up and going into the lumen of the stomach, so into the hollow part where your food is. We're going to pass through the neck cells here, right? So the neck cells secrete this mucus. And the reason for that is, right, stomach acid is very acidic. So it's going to break stuff down, and we don't want it physically breaking down the lining of your stomach, right? We want to protect these cells. We want to make sure that we're only digesting our food and not ourselves. So that mucus is helping us out as we come out of the gastric pits to circle around with our food. In fact, the stomach is so acidic, it's down to a pH of about 2. That's quite low. Now, this is necessary for a couple of reasons, right? One is the pepsinogen and protein digestion piece, right? So pepsinogen is in this inactive form, right? We need to activate it, right? So it's an inactive enzyme, basically. Right? And one of the ways that you can change protein shape is by changing the environment they're in, right? We learned about how proteins fold into specific shapes so that they come into contact with specific things and react, right? So if you change the pH of an environment a protein is in, that folding is going to kind of change too. So we activate pepsinogen in part with this super low pH in the stomach, right? In general, this acidic environment denatures proteins. When we say it denatures proteins, what we mean is it's kind of unfolding them for us, right? So it's reacting with our proteins, and it's starting this breakdown process by taking our proteins from this complex 3D shape, and we're starting to unfold them or denature them into their long polypeptide chains, so that then when we try to actually absorb our amino acids, right? We've got a polypeptide chain that we can use enzymes to chop up into chunks, right? So we're denaturing these proteins in the stomach with this acidic environment. Other useful function of the acidic environment of the stomach is that lots of things that can survive in the world around us, which is not a super acidic environment, aren't going to be able to survive in a fluid that's at a pH of 2. So you will still sometimes get food poisoning, right? You might not get totally lucky and have this low pH kill off all the bacteria in something you ate by mistake, but we can kill off some stuff, right? So this helps us kind of as part of our immune system, just this physical property of the stomach. Yep. Which means we got to make sure that we have this gastric mucosal barrier. So we just mentioned that the neck cells are secreting mucus right at the top here of the gastric pits. Right. We also have a continuation of the mucosa along basically all the tissue, the inside of the stomach. And so that's continuing to secrete mucus, and we're also secreting some bicarbonate here. So we've got this mucus mixed in with bicarbonate. So if you've got these protons, these hydrogen ions, this acid coming up close to the wall of your stomach, hopefully the mucus is helping it not actually touch our tissue, our cell layer itself. And as this acidity comes into contact with the mucus, there's some bicarbonate in there, which we know can buffer out acidity, right? So that's coming from those neck cells and from goblet cells, which are a little further up. Okay. So we've got our chyme in the stomach. It's this mixture of gastric juices with some enzymes, but also this acidity. We've mixed it in with our food that's all kind of chewed up. We're then passing it through our pylorus and through the pyloric sphincter in little bits until we go into the small intestine, right? So the point of the small intestine is really absorption. All the absorption, or most of the absorption of your nutrients is going to happen in the small intestine. It actually happens pretty fast. So you have extra space in your small intestine, essentially as kind of backup for this absorptive process. The absorption is usually happening in just the first 20% of the length of your intestine. Okay. Which means that if you have a surgery that takes out part of your small intestine, right, you still got some of this absorptive capability. Okay. When we zoom into the small intestine and look at the cell layers that are lining the lumen, we're going to see we have an anatomical arrangement that helps us even expand the surface area for absorption even more. Okay. So overall, we've got this coiled up hollow tube. You've seen it in lab. We call it the small intestine because it's smaller in diameter when you're holding it, at least usually, about 8 to 10 feet long. It's where we are absorbing nutrients and also while we're continuing to digest our food. So continuing to break it down until we've got little pieces that are small enough to pull through our cell layers. And we got the sections, right? So right after the stomach, we have our duodenum. It's relatively small. It's kind of C-shaped, curves back this way and a little bit into the posterior wall of your abdominal cavity. Then when we come out, we're in the jejunum, right? And we go along and along and along. We can just follow somewhere. I'm not going to follow these lines in our cartoon, but we're eventually going to find ourselves in the ileum, right? So the ileum is the last stop before we pass into our large intestine, which we also call the colon. Okay. So we're going to zoom into the wall of the small intestine now, right? So we're taking a little chunk here and looking at those cell layers. So we can see our mucosa, our submucosa, our muscularis externa, and our serosa here. Okay. And what we're going to notice when we look at this first layer, this innermost layer, the mucosa, right, we can see these big projections. Okay. So these big projections are called villi. Okay. So these villi mean that with this kind of fuzzy inner layer, we've got a lot of extra surface area here, which means we've got a lot of surface area where we could bump into an enzyme embedded in a cell wall, or a cell membrane. Sorry, not a plant, not a cell wall. Can't say that. Right? So we could bump into a cell that has an enzyme on it, or you could bump into a cell, and if we're thinking about a nutrient that's small enough, we can get absorbed, right? So that's part of why the surface area part is important. If we were to zoom even further on an individual cell, we can see that we have the mini version of those villi as well on each of these individual cells in this layer. So there are microvilli forming our brush border, so like a brush across. Yep. Now, what we might also notice, right, is that we've got some stuff inside our larger villi, right? So we'll talk about the details of some of what this is as we go through absorption. But notice that we have blood flow going into the villi, so the larger structure, right? So we've got venules and arterioles here. We got capillaries. And this green thing in the middle is something called a lacteal. So that's going to be part of our lymphatic system. And we'll talk about why that's important here, specifically in the small intestine, when we think about how fats get absorbed, because fats are going to work a little differently than everything else. And between those villi, we also notice that we've got these depressions, right? We've got these holes kind of dug out. We refer to them as crypts. These are our crypts of Lieberkühn. Okay. So the point of these crypts of Lieberkühn is that they have epithelial cells in them that are secreting a bunch more bicarbonate, basically. Okay. This is particularly going to be true in the proximal small intestine. So remember, proximal, proximity means near, close. So we mean the first parts right after the stomach is where we're secreting this bicarbonate-rich fluid. And you can think about why this is important as we think about that acidic chyme coming out of the stomach. We didn't want that acidity to burn the walls of our stomach. We also don't want that acidity to burn the walls of our small intestine, right? So we're buffering it out as we pass this acidic chyme into the small intestine. We do want to reuse that bicarbonate. So as we move into the distal portions of the small intestine, right? So the proximal most part would be duodenum, distal most part is when we get to the ileum. We're going to absorb that bicarbonate back so it can get reused by your body. Okay. When we're thinking about this proximal most part of our small intestine, which is the duodenum, okay, it's linked up to a bunch of our glands and the accessory glands of our GI tract. So we'll zoom in on the pancreas and the liver in a second, but we're going to see that they're basically delivering their products into the duodenum, right? So they're going to have tubes that pass into somewhere here. Okay. So when we think about the function of the duodenum, it's important to think about what those glands are doing too. So we'll think about the pancreas first. So the pancreatic juice is going to include a lot of different digestive enzymes, and we'll list them out specifically both as we go through the pancreas and then as we go through how we break down each type of macromolecule. But we've got that part, lots of digestion, right? Which hopefully makes sense as we think about, right, our chyme. All we've done is kind of denature some proteins and maybe start to break them down a bit. Right? As we pass into the duodenum, into the small intestine, that means we have to keep breaking down our larger molecules so that they're small enough and we can absorb them in that first 20% of our small intestine. And the other thing we have is even more bicarbonate. So bicarbonate from the pancreas as well is helping us neutralize that acidity of the chyme. We're also going to have bile coming down from the liver. Okay? So as we're going from the liver, potentially from the gallbladder if we're using stored bile, this is also going to dump into our duodenum. Now, this is going to contain, again, some more bicarbonate, but also very importantly, our bile salts. So when we think about fat digestion, bile is going to be particularly important, because these bile salts are going to help us basically break apart fat globules so that we can absorb them. Okay. So looking at the liver, since we just mentioned it, and also since it's the highest one up, our liver is large. You've seen it in lab. You've seen how it's attached to a lot of different blood vessels. And it has this portal system it's attached to. Okay? So essentially what happens when we're thinking about the liver's relationship to the rest of the GI tract is, right, we've got all these veins linked up to your small intestine, also linked up to your colon, right? Linked up to everything in here. And all those veins go back to the liver before they go back to the heart or anywhere else in your body, right? So this means that anything that you absorb from your food, right, when you absorb it, it doesn't go into an artery, it goes into a vein, right? Because we're going back to the heart. So we absorb stuff into veins, and all that blood, before it goes back to your heart, goes into the liver, where the liver can modify it, right? So the liver is kind of protecting you from whatever you happen to absorb, whatever you swallowed, right? So it can detoxify substances. That's one of its important functions. Right? It's like your filter here. So we're detoxifying stuff. And it also helps process some of the nutrients. So it can add stuff on to the little pieces that you're absorbing, combine them, kind of adjust the composition of molecules before we send them back to the heart and then around to the rest of the body. So that's really why we have this portal system here. A reminder, portal system really means that you've got extra kind of capillary beds linked together. So it's called a portal system because of all these capillaries. Okay. So we've got that vasculature attached to our small intestine, all the parts of the intestine, really. We're taking all the blood back to the liver before we send the blood anywhere else, and we're adjusting the contents. Okay. If we wanted to name the vessels, we've done that in lab. We have our different mesenteric veins, especially, right? We had our superior and inferior mesenteric veins heading back to the hepatic portal vein, right? So that big one heading into the liver. We can zoom in on what's inside the liver in a second, but we'd make our adjustments here. And then we would head back to the heart through a hepatic vein that sends us to the vena cava, which lies kind of through and just behind the liver. When we think about what other blood goes to the liver, right, usually we don't think of veins as going to somewhere because we've got deoxygenated blood in them. And the liver is special in that way that we do have veins going to it, but that's because it's processing the stuff that's in those veins, right? That's not where it's getting oxygen to feed liver tissue, grow new liver cells, that kind of thing. So we do still have an artery going here. We do still have a hepatic artery that makes sure the body of the liver is able to work as well. Okay. Then we got our liver up top. Ooh, something's weirdly out of order. Okay, well, we'll get back to it. All right, so we talked about our small intestine and our absorption. So if we're following our ball of food, you swallowed it through the mouth, down the pharynx, down the esophagus to the stomach. We went through the duodenum, where we had pancreatic juice coming in. We had bile from the liver coming in, passed through after the duodenum, the jejunum and the ileum, and now we're entering the large intestine, starting with the cecum. Okay, so our large intestine has cecum, then colon, which has 3 parts, ascending, transverse, 4 parts, descending, and sigmoid, and the rectum, and then the anus. Okay. And we'll walk through the different parts. Okay. So as we pass into the first part, right, so we're going to go through the ileum, through the ileocecal sphincter. right? So ileocecal, anytime you see that, it's just telling you it's joining the ileum, ileo, and the cecum, cecal. Okay? So the first part of the colon we enter is the cecum. Okay. So from there, we go through the ascending, transverse, descending, and sigmoid colon. And our overall goal is to concentrate stuff, right? So our chyme that we are passing into the small intestine, pretty watery, pretty fluid-like. So by the time we get to defecation coming out of our anus, right, we want that waste product to be relatively solid, okay? So to do that, we're going to be absorbing a bunch of water in particular, and we're going to store feces until defecation, which is hopefully voluntary. Okay. So when we get to the rectum and anus, so rectum, the straight up and down part, anus leaving the body. So the colon's dumping into there. We're going to pass through two sphincters before you leave the body. So the sphincters are on either side of the anus. We consider them part of the anus. The internal one you pass through first if your food coming out is made of smooth muscle. So that smooth muscle part means that that part's not really voluntary, right? Smooth muscle doesn't have somatic innervation. You don't control it with your brain, but you do control the external anal sphincter. So the external anal sphincter is the one that you can actively, deliberately, voluntarily control because it's made of skeletal muscle. Okay. So in order for our waste to come out, right, both of those need to open up, both that internal involuntary one and that external voluntary one for defecation to happen. Okay. So back to our accessory structures here. So we're going to think more about glands, right? So our general structure of a gland is going to be a body of the gland where we secrete a bunch of things into these structures called acini, these kind of blind ends or kind of bubbles. And then we're going to go into a series of ducts that lead us into the digestive tract. So we see glands in several different places. So you have the salivary glands, so in basically dumping into your mouth, right? And as food passes, pharynx, esophagus, stomach, duodenum, we're going to have the pancreas dumping pancreatic juice into the duodenum. And then eventually, and then about at the same time, we have our liver secreting bile also into the duodenum. And each of these is going to have probably acini in them, but really we mostly think about those when we think about pancreas, I would say, is the times we really cluster in on that little structure. Okay. So we're going to start at the top with our salivary glands. Okay. So our salivary glands, which you've hopefully seen and remember from lab, are producing some enzymes, some mucus, and a bunch of bicarbonate ions. So here we can see our parotid gland located kind of just below your ear. We have our sublingual and submandibular glands below the tongue and below the jaw with all their little ducts going into the mouth, helping you salivate. Okay. So in terms of enzymes that are present in that saliva, we want to remember the salivary amylase. So this is going to come back as we think about particularly how we digest starches. And we also have something called lysozymes. So that's our other lysing agent, our enzyme here in the mouth. Okay. So as we pass down, right, all the way past the stomach, here we're looking at where our pancreas and our bile ducts dump into the duodenum, looking at the pancreas first, right? So our pancreas has this head and then this body kind of pointing out towards the side, towards your spleen. We've got cells here that have both exocrine function and parts that have endocrine function. We actually talked about the endocrine function before, right? So the endocrine function, hormones, we're thinking insulin, we're thinking glucagon, right? Exocrine is mostly what we're focusing on now. So exocrine is where we're going to have all kinds of enzymes, all kinds of digestive stuff. Okay. So when we say that we're producing pancreatic juice, we're really focusing on that exocrine portion of the pancreas. So that's going to be coming from these cells in the acini, so our acinar cells, going into ducts that secrete a little bit of bicarbonate as well. Okay. So pancreatic juice, when it gets into your duodenum, lots of bicarbonate in it, lots of protein in it, because enzymes are proteins, right? So by the weight of the pancreas, it's actually making more protein than anything else in your body because its job is to make all kinds of different enzymes. So we're going to be thinking about pancreatic amylase, which is going to be important for carbohydrate digestion. We're going to think about lipases. So notice this lipe beginning tells you it's going to be breaking down lipids, right? Ase ending tells you it's an enzyme. We're going to have a variety of proteases. Proteases are enzymes that break down proteins. There are also nucleases here. So nucleic acids can also be broken down. Right. Mention the liver, secreting bile. Right? So we can see our hepatic bile ducts going into the common bile duct, which you saw in lab. We're connected to the gallbladder through the cystic duct. That's our kind of storage area for extra bile. If you're not eating, but the liver is still producing bile, that's where you put it. Right? When you're ready to use the bile, comes out through the common bile duct, which is joining up the same spot where the pancreatic duct is coming in. So we've got the structure called the ampulla of Vater. That has a sphincter called the sphincter of Oddi around it, letting us control the release of both the bile and the pancreatic juice into the duodenum. Okay. So in the liver, in addition to secreting the bile, we're processing nutrients we know, and we're detoxifying stuff that's come into the blood. Okay. A little more detail here on processing nutrients. We're going to be like kind of adding levels of detail as we go through different parts of this unit. Right? One of the things that we're doing is we're taking glucose and turning it into glycogen. Right? So glucose, our familiar friendly sugar. Glycogen is a storage, basically a stored form of glucose. So lots of glucose all linked together so that you can save it for later. We find it in the liver. We find it in some muscle tissue. We're taking amino acids coming back to the liver and transforming some of them. We'll look at how these link up with what we do with our fatty acids as well. We're going to make some triglycerides, we're going to make some cholesterol, and we're going to make some lipoproteins here in the liver. Okay. Liver actually does a ton of stuff. So in addition to that, we want to remember that the liver was involved with our erythrocyte life cycle, right? So our life cycle of red blood cells. So it was involved in that process where we're removing our old red blood cells. And particularly, we were paying attention to how the liver catabolizes our hemoglobin, so breaks down hemoglobin so that we can rescue that iron in the center of the heme group and reuse it. And it's generating bilirubin as a waste product. So the other parts of our hemoglobin we can get rid of, goes into your waste and eventually is eliminated as feces. Okay. That was another thing that we talked about when we talked about the blood. We also mentioned some of our plasma proteins when we're talking about the blood. So all our clotting factors are coming from the liver, right? So it's making that ability to do hemostasis, that ability to keep your blood in your body if you get a cut. And it secretes and modifies some hormones as well. Okay. So the liver, that big part, is making the bile. The gallbladder right next to it is its own organ. It's not making bile, but it's storing bile. So it's part of our biliary system. Our common bile duct links them together, right? Sending all that bile wherever it happens to be coming from, linking up to our pancreatic duct and sending everything mixed together to our duodenum. Right? So mentioned them when we saw them on the image, but here we see that pancreatic duct Here we said that ampulla of Vader. An ampulla is just kind of like a swelling, right? Like a hollow swelling. So what we're really saying is just this tube gets a little wider here, right? The ampulla of Vader before we pass through this sphincter of Oddi so that we can make sure we're only releasing this stuff into the duodenum when we actually have food to digest and absorb there. Otherwise, kind of no point. Okay. So here's the promised zoom in to the liver. All right. So in terms of what you want to prioritize for studying, I wouldn't worry too deeply about this stuff, but I know you probably didn't see much of this when you were looking at the liver in lab. So if you're curious about how the liver is doing these things, right, you'll notice we have these veins coming through the hepatic portal vein. That's one of the things that we have here, right? And as we come into the liver, we've got these kind of hexagons here made up of our different hepatocytes. So hepatocytes, hepato is just a prefix that refers to liver. Cyte is a portion of this word that means cells. So liver cells in Latin, basically. Right, so these liver cells are making bile. So they've got enough room to pull stuff up out of the blood into these structures called sinusoids to make the bile. We got arteries, right, giving the liver oxygen. And we've got this bile system, even at the kind of microscopic level. So here we see our little bile canaliculi that go into our bile ducts, and then the bile eventually drains out of the liver. Cool. So I'm also just reminding you here that we got a big table in your textbook that runs you through all the organs that you're interested in the GI tract and what they're doing, right? So what their job is. So as you're studying, I would kind of take a look at that as you're kind of focusing on what you want to get from this part of the slides. Okay. So we're gonna begin thinking about absorption next. I think this is gonna really tie together these pieces a little bit better. Okay. So we're gonna go through specifically how we absorb carbohydrates, proteins, and lipids. We're not really gonna talk about nucleic acids. It's only 3 of our 4 macromolecule types. And we'll talk about all the enzymes that are involved and where they're coming from as we do it. Okay. So carbs, proteins, and lipids we're going to go through, and then we'll have a brief mention of vitamins, minerals, and water. We're going to start with our carbs. Okay. So when you're eating carbs, right, you're eating a burger and fries, right? A lot of the carbs that you're going to eat are going to be in the form of starch. So we've shown that here, right? So if you're eating bread, potatoes, whatever, right? The animal form of storing, the plant form of storing glucose is starch. So it's the kind of homologous structure to glycogen in an animal. Okay. So starch is kind of interesting because it's not one big straight line. It's got all these branches, right? So we've got glucose monomers, but we also have this branching structure in a starch. This is going to present a little bit of a problem for us as we try to digest a starch, because you can only get a monosaccharide into your cells, right? So we need to break it down into just one monosaccharide before we do the absorption part, okay? So we're going to have to break up our polysaccharides, right? So we're going to have to chop up the starch. And after we chop up the starch, we'll find that the products that gives us after the first step are going to be these disaccharides. So 2 little carbohydrate monomers stuck together, and these limit dextrins, which are basically the branches, these corners that we couldn't get to. Right? And ultimately, you're going to have to break those down a little further into a monosaccharide to pull it into your body, send it into the veins, going back to the liver, and eventually back to circulation. Okay. So what are we doing? Where is this happening? So we're going to start in the mouth. If you've ever I don't know, held a cracker in your mouth a little too long, I feel like My friends got obsessed with this in like second grade for some reason. You might have noticed it turns a little sweet immediately. This is because starch digestion actually starts in the mouth, both from the chewing level, right? So that mastication physically breaking stuff up, but also the enzymatic level, right? So in the mouth, in your saliva, you have this salivary amylase that immediately starts to break down the starches, right? So we start to break down starch with that salivary amylase into disaccharides and limit dextrins, right? We're not ready to absorb them yet as we pass through the mouth, right? But we're part of the way there, right? So we're going to pass these disaccharides, limit dextrins, and any of the starch we didn't get to yet, right? Like you've still got chunks of food after you chew, probably just smaller ones. So not totally done breaking down our carbohydrates yet. Pass down your pharynx, esophagus, stomach, you get into the duodenum. Pancreatic duct is going into there. So pancreas also produces amylase. So we also have pancreatic amylase, continuing this breakdown of large carbohydrates like starch, giving us these disaccharides and limit dextrins. And then as we go to absorb these things across the walls of our GI tract, we're going to have a couple additional enzymes kind of embedded that break them down a little bit further. Okay. So we don't have a picture of those ones acting, but they basically be in the small intestine. Okay. So relatively short explanation of what we're doing with our carbs. Proteins are going to be more complicated. So we'll do a quick intro right now to our proteases. All right. So we're going to go through all kinds of different enzymes that break down proteins. And we are going to be thinking more about their details than we're thinking about the details of enzymes that break down carbohydrates. So we're going to see ones that are termed endopeptidases. So these are going to break apart polypeptides from basically their inside bonds. So we mean not the ends. Exopeptidases come and just pull off our last amino acids, right? So from one end of a polypeptide. And we're also going to have to think about zymogens. So this ogen ending, we've seen on all our inactive forms of enzymes is linked up to this zymogen idea. This is like basically a lump term for our inactive proteases. It gets stored in granules that we secrete, and then we eventually activate. Okay, so we'll go through the details of all of this on Wednesday, and we'll stop there for right now.